Today, we are talking about the application of machine learning in organic synthesis. We are also going to discuss robots in chemistry and some applications of deep learning in drug design. And before we begin, I would like to mention that today's lecture is more of a review type, in a sense that we aren't going to discuss many technicalities related to case studies that we are going to highlight today, just because understanding all technicalities of some works, of these works, would require a course in its own. So let's discuss, first of all, the importance of organic synthesis and what basically it is. Many chemists have a general understanding that organic synthesis implies making organic reactions in a sequence to get some target molecule. Basically, as an action, it is. However, organic synthesis is also, as a discipline, is also a field of study that deals with organic methodologies and some problems of organic synthesis design, some of which we are going to discuss today. Speaking of the importance of organic synthesis, we can definitely say that it's widely used, and it's a foundation of our culture, of industry, of many industries today. So organic synthesis are not only the basis for some fundamental research, it's the basis of medicinal chemistry, plastics production, cosmetics and perfume production, etc. So, of course, we, considering these different fields and different industries, we should say that organic synthesis of plastics and organic synthesis of medicinal chemistry, of drugs differs and may differ significantly, at least in its scale and its methodologies. But despite this, for today's purposes, we consider every synthesis, every sequence, every organic synthesis as a synthesis we are interested in. So a synthesis that can be predicted with some machine learning models. And currently, organic synthesis as sequences of reactions can be really complex. So just imagine, first we can remember one of the first successful and commercially used organic synthesis, the synthesis of aspirin proposed by Hoffman in the late 19th century. However, today, the molecules, organic chemists, and many people from industry are interested in are much more complex than this molecule, the molecule of aspirin. So just imagine synthesizing these molecules, and it can be a really complex task. And indeed it is. So today organic chemists often discuss, and some of them work in the field of total synthesis, which, and if we consider a total synthesis, it is a complete organic synthesis of a complex molecule, which is usually a natural product from readily available substances. And ideally, these substances should be commercially available on a large scale. So if we, for example, consider the molecule of Taxol, which is a chemotherapy drug, it's this molecule first is really complex, and next, this compound is, while being natural, so it's can be found in the bark, or in Russian, Korea, in bark of Pacific yew. So we need to process a lot of its bark to produce only 300 milligrams, or 1 dose of Taxol, which is also, as you can see, called paclitaxel. So it's known to be active in treatment, to be potent in treatment of several types of cancers, like ovarian, breast cancer, lung cell cancer, etc. So a lot of Pacific yew should be processed to synthesize, to obtain only 1 dose of it. And in the second part of the 20th century, proposing a synthesis of Taxol was a very relevant aim. And in fact, a synthesis was proposed. In fact, several approaches to its total synthesis were proposed in the last decade of the 20th century, and the famous synthesis of Taxol by Nicolau and his team consists of tens of organic reactions, and it's a really complex sequence in which every reaction is complex in its own. So proposing such a synthesis is a work of tens of people for several years, or maybe even for a decade. So it's a complex task. And there are several approaches to simplify total synthesis. We should mention, before we discuss approaches that utilize machine learning, we should mention several approaches that are used to analyze and propose synthesis, in other words, approaches to synthesis strategy. So the first, we should mention retrosynthetic analysis, which was proposed in the synthesis of this compound by Elias Corey. And you can grasp what retrosynthetic analysis is by looking at this scheme. So if we want to arrive at this target molecule, then we can analyze its synthesis backwards. So organic chemists could say that Wittig reaction involving this molecule would precede this target molecule, and this can be obtained by aldol condensation, and this one can be obtained by Michael addition. So in such a way, we analyze synthesis, and such an analysis has one particularly important feature. So we reduce molecular complexity stepwise, and in every step, we can propose alternative approaches to the synthesis of T1 or T3. So we approach the synthesis in a modular way. And predicting organic synthesis is a difficult task for machine learning or deep learning. Consider, for example, a hypothetical machine learning model that is correct in 90% of cases. So, after all, it has only a 1.5 chance of getting 18 predictions correct in a row, if we assume independence, of course. And having this in mind, we can conclude that synthesis prediction is a complex task both for humans and for machine learning models, yet it's now solvable. The second synthetic strategy we should mention is the concept of convergent synthesis. So if we combine organic reactions in a linear fashion with yields equal to 80% in each time, then we will get only the yield of 11%. So it's basically 0 0.8 power 10. And we can approach the synthesis in a convergent way. So we first can synthesize C in 3 steps and D in 3 steps as well. So combining these precursors and continuing synthesis in several steps will get us target molecule with the yield of 10 to 1%. So it's better, of course. However, we can engineer the point of convergence later, and we can get the yield of 41% if we delay the convergence point as late as we can. So this allows us to approach every stage modularly, and in case any difficulties in synthesizing C, we can change this sub-synthesis as needed. So this also relates to the possible accuracy of proposed models. So we can train a model in such a way that it proposes convergent synthesis, and in such a way we can get syntheses that are more reliable. So what should we discuss? I propose to discuss one of the works on synthesis prediction, one of the recent works from 2020, and this work is the development of the research project we had discussed previously, implemented in the IBM for chemistry platform. So this work can be easily accessed on the journal site, and it's open access. So considering this work, it's in fact highly complex, so the authors outsourced technicalities to the supporting information, so you can download it as well. So it consists of more than 200 pages. So, however, we are going to discuss briefly the approach proposed by the authors, and we are going to I'm going to demonstrate the result, its result of the corresponding platform in action. and you can do it at home as well. So synthesis prediction is a task of increased complexity. So for such a task, as I will show later, so for it, there should be no reactant reagent discrimination. And in such a way, reactants, solvents, catalysts, etc. are considered as precursors. So for synthesis of this polycyclic molecule, we need to consider all these substances. And the authors propose to consider synthetic roads as hypergraphs. The concept of hypergraph is an extension in which edges hyper edges may connect any number of vertices in a given graph. And this allows us to have edges that connect three edges in such a way, or we can even have loops, etc. So these synthetic roads get very complex in very rapidly if we consider organic synthesis. So we need to arrive from commercially available reagents to the target molecule. So if H is a target molecule, we may have such a hypergraph that encodes the synthesis of H from I, L, A, which may also be commercially available, and several other commercially available compounds. In such a way, K is a byproduct that isn't used in the later synthesis steps. So as an example of synthetic roads, of alternative synthetic roads from the data set used by the authors, we can analyze the synthesis of 5-bromomethoxypyridine, and it can be synthesized in several ways from several precursors. So all these one-step synthesis may be included in a synthesis of some compound for which this one is a precursor. And the workflow for the prediction of retrosynthetic pathways is the following. So we can select a target molecule for which a retrosynthesis is proposed based on encoded in in the model. So this retrosynthesis implies several precursors which are analyzed to be canonical smiles and to be unique. So these precursors are analyzed and analyzed in a way, in the following way. So first, to which reaction classes we may assign individual reaction in a retrosynthetic pathway. Compounds are also scored on the by the so-called synthetic complexity score. You can refer to this work, that is, for which I included a link. So the synthetic complexity score correlates molecular complexity with the expected number of reaction steps. Indeed, if some compound, if a synthesis of some compound requires great number of synthetic steps, it's complex. And so indeed, synthesis of Taxol, which is molecularly complex, requires a lot of steps. And so for every reaction, a model for forward reaction prediction is applied to estimate confidence in a particular step. So low confidence reactions are filtered out, and high confidence reactions are used to update our reaction hypergraph, which is analyzed to start from commercially available precursors, and if it is, so we get a retrosynthetic scheme for our target molecule. And if no, the cycle continues. We can get a glimpse of how this model works by using the site we have discussed in the previous lecture. So while previously we discussed the forward reaction prediction capability of the IBM RxN platform, then today we are going to discuss predicting retrosynthetic pathways. So let's specify a target molecule that can be aspirin, which would be really simple. Let's select this compound, allicin. for which we can get smiles from Wikipedia and input the visual editor. So we get visual representation, and next we can select several models, from which let's select this one, and we can select automatic mode. We can check, we can select quality. We can customize retrosynthesis by selecting some starting materials that are available, and we can submit a job. The job is processed on the server, so we will need to wait a little bit. And before that, we can analyze some proposed synthesis. For example, the synthesis of aspirin can be really simple, and I would say that it's a suboptimal synthetic pathway, although it may be really convenient for execution in a lab. So we can see that some precursors are more complex than the target molecule. And in fact, there are a number of synthesis that was proposed for our target molecule, and some syntheses are really meaningful, while others are indeed suboptimal. So for every project, we get a history of submitted tasks, and now we got our results. So we got results for our submitted job. So we can get this compound in a number of simple and more complex reactions from commercially available reagents, while some are commercially available. So for this compound, several one-step syntheses were proposed. So these sequences differ in confidence and differ in the set of proposed precursors. So you can analyze all the proposals. A two-step synthesis was also proposed. And we can also analyze job I have submitted earlier today, and this is a job for retrosynthesis prediction of taxol. So there are several proposed sequences, and you can note that while these sequences start from commercially available substances or substances that are really simple and produced in en masse. So some complexes or some substances, while being commercially available, are quite complex. So it's not a total synthesis per se, or total synthesis as we expect it to be. And as you can see, all these sequences has lower confidence just because confidences for every steps accumulate, and we and it's really difficult to get a confidence close to 1. So IBM RxM works in this way, and it can be used for several other purposes as well. So I suggest you to visit this site and tinker. And now we are going to discuss our next topic for today. So let's talk about robots in chemistry. And first of all, we need to discuss why should we even use robots. And let's go back to the first lecture in this course. So as we have discussed, we in science, when we plan experiments, we expect some results. We expect to synthesize some new materials, or we check some models or theories, and we expect some results based on our previous knowledge. However, experiments sometimes just doesn't work. And such a state was called a cloud by Professor Uri Allen. So it's a state when you conduct experiments and experiments don't work, experiments don't work, experiments don't work. And unexpectedly, you get unexpected substances, materials, models, theories, etc. So this is viewed as a way to truly innovative knowledge just because we expected something and got something really novel. And in the framework, in the theoretical framework of the rational design based on machine learning, we have the following schema. So we try to combine scientific data, data from databases, literature, and lab notebooks. Don't underestimate lab notebooks with machine learning models. So we aim to propose new substances, materials, drugs, etc., with optimal properties. And we aim to save money, time, and potential environment. However, in this schema, we get some substances or materials, whatever, that are in fact expected. But can we get something unexpected by applying machine learning models? And this can be done, in fact. However, what we need to do is first conducting experiments in some way, and second, we need to some new approach. And solutions to the problem, or I would say ways to our goals, would be to use some robotization. And there are basically two equivalent approaches that are really interconnected, but they operate different terms. So we can speak of self-driving laboratories, and we can speak of the computer. So you can refer to these works, and let's discuss the foundations of self-driving labs or the computer. So first of all, let's consider a so-called closed-loop controller. So if we have some signal that deviates positively or negatively, we can measure an error or deviation and input it into controller. So controller gives a signal to the system that corrects the output signal, and it can be analyzed with some sensor in such a way that we can compare it with the reference and estimate if we have a positive or a negative deviation. So we get closed loop. And second, we should use high throughput experimentation to get efficient self-driving laboratories or some chemical robots. And let's have a quick recap. For example, we can, use such a platelet with many catalytic reactors, and we can conduct catalytic experiments in parallel. And in such a way, we can vary many different reaction conditions and ligands and search for combinations of reaction conditions and ligands that allow us to get the highest yield. And this closed loop approach is indeed the foundation of self-driving labs. We can use some machine learning models and some software to predict experimental conditions that should work best. Then we use some automated robotics platforms to perform an experiment, to make some measurements. And this data can be used to extend our data sets that are used to propose new conditions, and in such a way we get a closed loop. Let's discuss the series of works that describe the development of the computer. So in the first work, the authors proposed such a system that was used to synthesize several organic molecules, and such an automatic platform is quite complex, in fact. So let's discuss its architecture. First of all, it includes several modules used to conduct a reaction, including a reactor, a filter. After a reaction, we need to separate reagents, and it can be used a funnel, or it also a rotary evaporator should be used. So all these modules are combined by a series of pumps, and this is the hardware that is used to conduct reactions automatically. It looks like this. So we have all this under a bench. So this is a device that can be let on its own to produce some synthesis. And this hardware can conduct a large set of basic operations like mixing, cooling. It can quench reactions, or it can filter post-reaction mixtures. So crystallization may be performed, etc. However, one needs to control all these operations somehow. And here, the software part should be discussed. So the architecture of the software part is quite complex. Given a reaction scheme, we can use the specifically designed XDL language to input the written scheme to the device. Then the chemical assembly code is used, and in parallel, the authors also proposed the so-called GraphML to describe the topology in a given operation. The topology of pumps, rotary evaporators, reaction vessels, etc. So all this is the input is the combined input to the so-called compiler that we will discuss a little bit later. So and on the interface level, we interface to the hardware level, we get some simple operations that control pumps, stirrers, rotary evaporator, etc. So first, several words on the chemical description language, or XDL. There are several specifications to it, and in the supporting information to this article, the authors give a lot more details to XDL. So this chemical description language is designed to be human readable in such a way that we can describe reaction schemes to a computer. And it also should be machine readable. So it's based on XML. There are also several constraints related to its usefulness as a control language. So it has to have a defined namespace, should be unambiguous, and it should be transferable to chemical assembly code. That's because this high-level description of an experiment should be translated to low-level language that is processable by the device. And so chemical assembly programming language, as authors call it, specifically serves this task, and it describes elementary basic operations like moving a specified volume of reactant, or, for example, controls the pump, or it can start stirring, etc. So we translate high-level experimental descriptions to low-level commands. And with the combination of GraphML, we get the compiler to decompose our description of an experimental procedure in such a way that is processable by the device, and so it can get exact commands for pumps, stirrers, heaters, etc. However, we need to get some analytics hardware to get a fully automated system. And an approach to this was proposed in the second work from this series. In this work, the authors proposed the computer as we discussed it, so it can execute experiments in parallel, and it includes analytics hardware as well, like mass spectrometer, nuclear magnetic resonance spectrometer, infrared spectrometer, etc. So this device can be used to perform reactions automatically and to explore reaction space in the following fashion. So we can combine different chemical compounds from a pre-selected reaction space and encode reaction mixtures with vectors. So after performing a reaction involving a selected reaction mixture, we can combine an obtained spectrum, let it be an NMR spectrum, with a theoretical prediction. And if we have no new signals, the reaction is classified as the mixture is classified as unreactive. Or we can get a case, we can arrive to a case of a reactive mixture if we get some new signals in the obtained spectra. So this is basically a closed loop system in action, and after a set of exploratory random experiments, we can take some combinations of reactants from the pool of starting materials, combine them, or the computer combines them. It analyzes the results by using a machine learning model and updates the reaction database. So on the basis of which we can the computer proposes new experiments. And the authors used programmed the computer to conduct a series of experiments involving different great number of reagents. The computer used linear discriminant analysis to pre-select reaction mixtures that are more reactive, and explored the whole reaction space. So initially, the number of reactive sequences was higher. Comparing a random exploration, on the second phase, the authors used the reaction space of the Suzuki coupling, and they combined different heteroaryl halides with different boron organic compounds to get a coupling product. Again, they encoded reaction mixtures as vectors. And after some exploratory phase, they got the computer to pre-select reaction mixtures that are more reactive or that are more yielding. So after all, they get a model that predicts reaction yields in the Suzuki coupling. And as the final task for the computer, they conducted some free reaction space exploration and proposed several new reactions. However, there was some criticism related to this part of the work that, however, doesn't negate the positive results after all. There is a review that I can suggest on the topic of automation in chemistry, and you can in which there are several related fields are discussed, like flow chemistry and specialized synthesizers, etc. Speaking of automation, there are several pros that are highlighted by the authors of these works. So it is believed that using these such automated approaches increases productivity and reliability by reducing human error and by linking software and algorithms, one can easily produce a graphical representation of the results. What I would see as a great feature is that in such a way we get increased safety because we can automate experimentation with some dangerous substances like toxic substances or potentially explosive substances. Possibly we can reduce waste, at least it is said so, and improve reaction outcome quality. What is also important in my opinion, and I totally agree with the authors, that we get more walk-away time for the chemist. So a researcher can focus on analyzing data and do more meaningful task instead of being at a bench for hours. And another important subtopic that we should discuss when discussing robot-assisted experimentation is the approach to assessing the novelty of the obtained results. So if we get some data on reactivity of some compounds, we should always ask if these results are repeatable. And if these results, if these experiments are irreproducible, we should, of course, consider this data invalid. And only then we should ask a question if this data is new. So if this data is unexpected, so it's novel data on reactivity or on some compound properties. And of course, we would like to get here with the robot assisted synthesis. And you can get general workflow here. So there are several proposals how the community can develop self-driving laboratories, and there are proposals to use cloud-based data storage and design online analysis platforms. So there are many ways to boost the research by using self-driving labs, and we will see what will come in the recent years, I guess. So let's, and now let's discuss a couple of case studies that I find exemplary on the use of deep learning in drug design. So first, let's discuss how deep learning approaches can be used to boost antibiotic discovery. And in the work published in the Cell journal in 2020, the authors proposed helitin as a new antibiotic. And here we should mention that this compound was originally proposed for the treatment of diabetes. However, later in clinical trials, it was found that it has some unwanted properties. And it was shown that helitin has wide scope antibacterial activity, including the activity on some antibiotic-resistant bacteria. So the authors used a neural network to pre-select a set of molecules from a chemical library, and the predictions of the model were experimentally evaluated, and the authors came up with the proposal of helitin. You can refer to this article for a detailed description of approach used by the authors. So they combined convolutions and descriptors with convolutions centered on bonds instead of atoms to describe biologically active molecules. They used a number of features to encode molecular structure. So in such a way, they got a model for which even small data sets were enough for adequate accuracy of prediction. And the second paper I would like to mention is the paper from 2018 from Journal of Chemical Information and Modeling, in which the authors proposed a generative model that generates basically drug-like molecules. And the authors used two data sets of drug-like molecules. First, they selected 15,000 drug-like compounds from the zinc database, and also they used a subset of 15,000 drug-like molecules from ChemDiff. The statistics on these data sets is presented, and what the authors did is that they designed the so-called rank model, which employs the paradigm of reinforcement learning. And in this model, two neural networks, generator and discriminator, competed in the following fashion. So they had data sets of drug-like compounds, and entries from these data sets were mixed with samples of generated molecules. These molecules were generated by the generator, and the purpose of the discriminator that included CNN was to determine if these molecules are real or fake data. And this model allowed them to generate drug-like molecules that do not deviate by their structural parameters from molecules from the original data set. The authors compared the results of rank with the results of the previous model called organic. And compared to this, rank allowed them to reproduce molecules with basically the same or quite similar length of the longest chain in the carbon framework. The authors compared structural parameters or properties like log P and number of atoms, and indeed they got a model that generates drug-like molecules that are quite similar to those from the original data set. The statistics on molecular weight and topological polar surfaces are also given. And indeed, they get quite similar distribution. But considering the rank model, one should ask the most important question: what type of activity do generated molecules have? And in fact, the model gives no answer to that. So rank proposes drug-like molecules for which biologic activity can be evaluated, but obviously it was beyond the scope of the study. So there are samples of generated molecules. And this is all for today. Thank you.